Vitamin E is absorbed together with lipids in the small intestine, depending on good pancreatic function and secretion of bile. Tocopherol esters, which are present in food and supplements, are hydrolyzed before absorption. Vitamin E is transported via the lymphatic system to the liver. Alpha tocopherol is the vitamin E form that is mostly present in blood and tissues. This is because a liver protein called alpha tocopherol transfer protein, alpha TTP, prefers to incorporate alpha tocopherol into the lipoproteins. These lipoproteins deliver it to the different tissues. The other vitamins that end up in the liver are not incorporated into the lipoproteins, but are rather metabolized and excreted into bile. Vitamin E is found in most tissues. The highest vitamin E contents are found in the adipose tissue, liver and muscles. The pool of vitamin E in the plasma and in liver turns over rapidly, but the turnover of the content of the adipose tissue is slow. The best established biological function of vitamin E is that of a fat-soluble antioxidant that prevents the propagation of free radical reactions. Free radicals are formed in normal metabolic processes, such as the release of energy from glucose and fatty acids, and upon exposure to toxic agents, for example cigarette smoke and pollutants. Free radicals are very unstable molecules and look to bound with other molecules. This may change these molecules, and this can lead to loss of function and tissue damage. Free radicals are free because they float around until they stabilize, and radical in the sense that there are a wide variety of molecules from which they can take an electron. The damage doesn't stop there, however, as the new molecule, say a polyunsaturated fatty acid, a PUFA, in the cell membrane is now also missing an electron and has become another free radical. It is especially this snowball effect that damages healthy cells and tissues. Here is where vitamin E comes into play. Vitamin E is located in cell membranes. It protects PUFAs and other components of the membranes from oxidation by free radicals. The inside of the cell membrane is not accessible to water-soluble antioxidants. The fat-soluble tocopherols are perfectly suited to protect the membranes from free radicals. The fat-soluble tail of the tocopherols can reach in and neutralize the free radicals. Vitamin E neutralizes free radicals by donating a hydrogen atom. When the tail of vitamin E is oxidized by a free radical, it moves to the surface, where the head of the vitamin E can donate hydrogen. In this way, vitamin E pulls free radicals out of fatty membranes for neutralization. This vitamin E cannot perform its antioxidant function a second time until it's regenerated. Vitamin E can be restored by vitamin C, which in turn can also be restored by other antioxidant molecules. It is important to realize that most of the studies that investigated the antioxidant activity of vitamin E have used relatively strong oxidants as the source of radicals. Studies in lab tubes with low concentration of oxidants have suggested that vitamin E may also have pro-oxidant rather than antioxidant action. Apart from maintaining the integrity of the cell membranes, it also protects low-density lipoproteins, LDL, in the blood from oxidation. LDL transports cholesterol away from the liver to tissues. There is a close association between elevated levels of LDL cholesterol and heart disease, which is a leading cause of death all over the world. LDL is not known to contribute to heart disease unless they are oxidized by free radicals. The creation of atherosclerotic plaques and its progression to heart disease is only partially understood. When LDL levels in the blood are elevated, the oxidation of LDL may lead to the accumulation of fatty slabs on the inside of the arterial walls. This is the leading theory of the development of plaques. The elevated levels of oxidized LDL stimulate immune cells to attack the LDL using free radicals. This causes more oxidized LDL. The immune cells become bloated foam cells, and these foam cells attach to the inside of the artery. This can then lead to blockage of the coronary arteries and a heart attack. Vitamin E protects LDL from oxidation. It's added to LDL in the liver by the before-mentioned alpha-tocopherol transfer protein. This train of thoughts suggests that vitamin E may protect against heart attacks. However, and I would like to stress this, large-scale clinical intervention trials with tens of thousands of patients at risk for heart disease provided no evidence at all that this is really the case. Recently, non-antioxidant function of alpha-tocopherol have been identified. Alpha-tocopherol inhibits a protein that's involved in cell proliferation and differentiation. Vitamin E also inhibits the blood clotting and widens blood vessels. This eases the passage of blood through capillaries and narrow blood vessels. Alpha-tocopherol has a regulatory effect on the tone of the muscles of the circulatory system. As the heart pumps, blood vessels must expand and contract. Alpha-tocopherol supplementation has been correlated with maintaining normal arterial wall flexibility. Alpha-tocopherol can also activate a nuclear receptor called PXR, analogous to vitamin A and D that activate RXR and the vitamin D receptor. And thereby, alpha-tocopherol is able to regulate the expression of target genes. But the metabolic significance of such activation is not clear yet.